आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Purvasha Banerjee and Shushanto Das.
एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़, की आवाज़।
Vigyan Ki Awaaz podcast series aims to popularize new ideas of science and technology, which highlight STEM, science, technology, engineering, mathematics, and medicine. This brings to the surface the efforts and researches of Indian scientists, which is desired to bring a change in community learning and teaching, access to knowledge of resources, and social paradigms. Our goal is to spread the name of indigenous Indian research stories and their masters among the masses of society in a very easy manner. Bose Institute was set up in 1917 by Sir Jagadish Chandra Bose, the founder of modern science in the Indian subcontinent. It is Asia's first modern research center devoted to interdisciplinary research and bears a century-old tradition. The institute was founded as not merely a laboratory but a temple, with its principal aims being the advancement of science and diffusion of knowledge. Swami Vivekananda, Sister Nivedita, whose influence is reflected in the institute's emblem of Vajra, was significant in the setting up of the institute, and Rabindranath Tagore were among those who had consistently inspired and supported Jagadish Chandra Bose in his endeavors. Ever since the establishment of the Department of Science and Technology, Government of India in May 1971, Bose Institute has been an autonomous grant-in-aid institution of DST. Over the last century, the institute has endlessly strived to preserve the legacy of its illustrious founder. In this episode of our podcast, we welcome Professor Basudeb Maji as he speaks about his works and shares his insights.
I am Basudeb Maji and I am a chemical biologist here working at Bose Institute. So I did my PhD from Indian Institute of Science, IISc Bangalore, and then I worked at Harvard Medical School and MIT for 5 years, and then I started my own lab, and it's been 3 years now. But here at Bose Institute, I'm working for last 1 year. As I mentioned, my research topic is on chemical biology, and it deals with disease biology, and I try to combine chemistry and biology for the development of new therapeutic avenues or interventions.
Professor Maji focuses on the various details of drug discovery which have immense impact of human welfare.
So I'll focus on the topic of drug discovery because it is the most fundamental requirement for our existence. If we cannot have potent drug, new drug, whenever it is required, then mankind cannot exist. So we need to have methods for developing drugs Now, let's understand the procedure of developing a drug molecule. So generally there are two methods. One is computational, which I'll keep it aside because I'm not an expert in that field. The other one is the experimental, where we either develop the molecule, develop the drug candidates in chemistry lab, or we isolate them from natural sources. And then we make library and then test them, test their efficacy, and then take it to the next level. Now, so basically we need to have enough drug candidates for testing. That is one of the bottleneck. And if you think about the mechanism of drugs in general, there are two different spaces. One is extracellular, that means outside the cell. For example, if you talk about vaccine development using protein, that happens outside cell. The other compartment is the cellular compartment inside the cell. There we mainly focus on small molecule drug candidates. So now small molecule drug candidates has many benefit over protein-based drug candidates because of their easy production, storage, and so on. If you think about small molecule drug development, again, you have to have them in large so that you can find out the most potent one. So our lab or my work is focusing on small molecule drug candidates in high throughput manner. So far, the high throughput way of developing drug candidate is high throughput screening. That means you need to have a library of thousands and millions of drug candidates, and then we have to screen them to find out the most active one. And that is super expensive, and that requires a lot of automation. What we want to do, we want to replicate the system inside cell. That means we want to run all this process of developing drug, screening drug, and then identifying the lead candidate inside cell so that we can cut down on requirement of facilities and resources. So now inside cell, if you think about drugs inside cell, there are three challenges. One is how to take the drug inside cell, that is called cell permeability. Stability of the drug candidate, that means how stable the drug is inside cell. And third is having low off-target activity, how precise the drug is. Now to integrate all these components and then developing a high-throughput platform for having a drug lead, we need few things. We need to have an engineered system, cellular system, which can make these drug candidates in diverse in number, and then also test their stability inside cell and activity inside cell and give us the lead candidate. So to integrate all these components, what we are doing, we are developing a synthetic biology-based drug discovery platform. Why synthetic biology? Because we use microbes like bacteria. So it's not normal bacteria. We engineer them in such a way that they will make drug candidates, and then they will also test whether the drug candidates are stable or not, and whether they have on-target activity with minimum off-target activities. And we can get the lead candidate. So this is the whole platform we are working on. Now, if you think about nature of drug molecules, so as we know, protein is one of the best candidates in vaccine development, but delivering a protein molecule to a cell is the most challenging task because of its gigantic size and its charge and so on and so forth, stability as well. Now, if you can make it smaller, you get to peptide level. Now, peptides are active drug candidates. For example, if you think about even vancomycin antibiotic, if you look at the basic of vancomycin drug, so the basic structure is actually amino acids. They're linked one to each other, and it's a peptide molecule. So we are currently focusing on peptide-based drug candidates. The most advantage is you can exploit cellular protein synthesis machinery. Every cells need to do protein synthesis for their survival and existence. So the system is already there. That means they are capable of making peptides. Now we have to make use of it to make our drug candidates. So we are exploiting the protein synthesis machinery of live cell and making the drug candidates. So we are mainly focusing on peptide-based drug candidates, and we are making use of synthetic gene clusters, which can make millions of drugs at the same time and test them in parallel. So basically, we are developing an orthogonal strategy or orthogonal method for antibiotics due to recent surge in antibiotic resistance global, and that's actually killing the activity of many antibiotic molecules. So over time, we'll see antibiotics are not going to work. For example, UN has already predicted by 2050, there might be 10 million deaths per year due to antibiotic resistance. So we do not take the challenge seriously and we do not develop orthogonal strategy. So human lives are going to suffer a lot. So to find out a way, so we are developing, it's called phage therapy or bacteriophage therapy, which is going to be orthogonal to antibiotic resistance, antibiotic-based therapy. And that uses another microorganism called virus, which is going to take on the infectious bacteria. So with our combining our drug discovery platform that is synthetic biology based and combining with bacterial virus, we'll be able to tackle AMR, antimicrobial resistance, and probably we'll be able to develop a strategy to take on the challenge.
Professor Maji explains the work with the help of examples so that all the listeners can better understand this intricate area of scientific inquiry.
So as I said, the main challenge in front of us is the superbug. That means the pathogenic bacteria who are becoming resistant over time against antibiotic drugs. So these superbugs, they have high immunity against both drug molecules as well as their infectious bacteria called bacteriophage. So we want to develop drug molecule which can actually break the wall of adaptive immunity in these pathogenic bacteria and make them susceptible towards infectious virus to them. So basically we'll kill their immunity. At the same time, we'll use bacteriophage virus to make them more prone to infection or more prone to death. So basically it's a combination of drug and biocontrol. Why biocontrol? Because we are using another organism to kill them. So it's a combination of drug and biocontrol to take the challenge of antimicrobial resistance.
We extend our sincere thanks to Professor Maji for sharing his knowledge and help us understand the significance of research in the field of drug discovery.